Last time on the last video I uploaded, we learned about how genome sequencing has revealed the DNA code. But now we have to figure out how the code actually works. Scientists have three main strategies for doing that. We're going to go through them one by one, so let's just get straight into it. Strategy number one, the genetic screen. This is the oldest of the three, dating all the way back to the early 1900s when people like Thomas Hunt Morgan were using radium to mutate little fruit flies. But we'll be talking about the modern technique. The basic idea is that you cause some weird physical mutation to happen to an organism, and then you look back at their DNA and figure out what changed. Let's go through an example step by step. Step 1. Pick an organism. I'll pick a frog. Ribbit. Step 2. Sequence its DNA to get a baseline. Step 3. Hit said organism with a blast of radiation and pepper its DNA with random changes. Step 4. Mate our frog with a normal frog and see if any of their kids develop any strange physical mutations. Ribbit. Step 5. Sequence their DNA again and find the spots where the radiation made those random changes. Step 6. Match up the DNA mutations to the physical mutations. Like, I'm gonna go out on a limb here and say that this bit of DNA probably has something to do with number of heads. The genetic screen is kind of like bashing the engine of a car with a giant wrench and then driving it to see what you broke. I think that pipe we hit may have had something to do with the steering. Strategy number 2, population genetics. Population genetics is when you look at the DNA of hundreds or thousands or millions of people and compare them using statistics. Like, do all short people have bits of DNA that tall people don't have? Do all people with green eyes have similar bits? Do all people with breast cancer have similar bits? Etc., etc., etc. Strategy number 3, comparative genomics. Comparative genomics is similar to the first two strategies in that we're looking for differences between different sets of DNA. But this time, we're comparing between different species. Imagine two species that are really, really similar, like a horse and a unicorn. The unicorn is exactly the same as the horse, except the unicorn has a horn. And it can fly and fart rainbows, but we'll ignore those for now. If you sequence the DNA of both animals, it turns out they're like 99% the same. Well then, it's probably a safe bet that the horn is somewhere in that other 1%. The only problem with this is that unicorns are imaginary. When scientists do this, they use real animals, like the Antarctic ice fish, the unicorns of fishes. If you're subscribed to this channel, you might remember when we went to Antarctica and caught these weird fish with no red blood. Well, it turns out the Antarctic ice fish are a really interesting group of animals for comparative genomics because they're normal fish, but they're also super weird at the same time. They're the only vertebrates in the world with no red blood. They have these insanely fragile, low-density bones. They have gigantic hearts. They have all these super weird features. And some of the features actually mimic human diseases. So comparing their DNA to other fish is a great way to start honing in on bits of DNA that might be linked to some of these individual traits. To summarize, the genetic screen is when we compare mutated versus non-mutated individuals of the same species. Population genetics is looking for patterns inside of the naturally occurring variation of a single species. And comparative genomics is looking at the differences between different species. So now what? We've identified these three interesting bits of DNA, number of heads on a frog, breast cancer in humans, and the unicorn horn. Was there any way to actually test these out? Like, if this is really the unicorn horn DNA, if we deleted it, the horn should, disappear, yes? Well, sadly, sequencing only works one way. Like, we can read the code, but we can't, edit it or change it. That only happens in science fiction movies where some evil scientist is trying to make super intelligent sharks or whatever. You can't edit DNA like that in the real world.
Oh, my sad, sad, naive friend. You really think DNA can't be edited in the real world? What, have you been living under a rock? You have obviously never heard of CRISPR.
Next time. I guess Pyramid Eye was right. We're gonna edit DNA with this new thing called CRISPR.